Hello everyone and welcome to chapter 2.3. This is proteins and you can see there's some cute eggs here. Anyways, yeah, we're talking about proteins today. And we always start off with the same thing, a few basic details. Number one, what elements is it made of? Carbon, hydrogen, oxygen, nitrogen. This is new. And sometimes it also has sulfur. Some amino acids have sulfur. Now, proteins make up a lot of structures, and these are some examples. Hemoglobin, which is found in large quantities in your red blood cells. Collagen, which is like in your skin, right, and your connective tissue. They also made up of components of cell membranes. Yeah, and these are like transport proteins at your cell membrane, other proteins as well. Enzymes, all enzymes are protein. Next chapter, we'll learn a lot more about enzymes. Just remember, enzymes are always protein. Antibodies are also always proteins. Antibodies are this molecule that your immune system produces to fight off the disease. And you can see how protein is so important in the process, right? Keratin, all keratin is made of protein. Keratin makes up your hair, okay, your nails. These are all made of keratin. So yeah, there are many, many more structures made of protein. This is just a limited list. These are just some examples. Now, what do we call the monomers? The monomers are called amino acids. The dimer is called dipeptide, and a polymer is called a polypeptide. Okay, amino acids. Amino acids are drawn like this. Now, amino acids tell you that there is an amine group somewhere, and this contains nitrogen, which is an element found in all proteins, right? There's an amine group, so NH2, and this carboxylic acid group is also present, C double bond O, OH here, and it's all connected to this central carbon atom. This central carbon atom is always connected to hydrogen and then a R group, which could be anything. There are 20 different types of R groups, which fortunately you don't need to know. You only need to know how to draw one or two of them. So yeah, this is the general structure of amino acids. I think I recommend you practice drawing. I always start from the central carbon. Then I draw the amine group. Sometimes I draw it like this, just NH2. Then I connect it to COOH. Sometimes I draw it like this too. Okay. See how it's connected in sequence. Okay. And then one hydrogen on the top, and then R group at the bottom. Okay, get used to drawing it. So yeah, that's the general structure of amino acids. You need to know how to draw it. Now, these are other versions of amino acids. So this is a skeletal structure, and you need to know how to identify this. But this is a little bit tricky because you cannot see the carbon suddenly. But you can identify the nitrogen, which is always drawn. So this is the amine group. And now you can see the carboxylic acid group, which is this double bond OOH things. And it has a carbon, which is right here. This is a carbon. And you can identify the central carbon. This is central carbon here. Central carbon at the center. The H is not drawn. And then there's an R group here. So yeah, if you are able to, find the double bond O and find the N, you should be able to find the central carbon with no problem. Now, this is another way to draw it as well. This is amino acids that are ionized in the state of water, ionized state in water. And this really shows you how amino acids can act both as an acid and as a base, because as you can see here, the H in the carboxylic acid group has dissociated, so it forms COO negative. And whereas the NH2 group, the amine group, has gained a hydrogen, so it becomes NH3+. So it's one side is negative, one side is positive. So overall, it's a neutral charge in general. But the side chain can have charge as well, which gives you many types of amino acids like this. So you can see here, there are positively charged R groups, negatively charged R groups. And if the R group is positive, then the whole thing is positive, right? And if it's negative, the whole thing is negative. Now, you do not need to memorize this. You only need to know these types of amino acids. Let me point it out to you. Number one is glycine. This is what you need to remember. You need to know how to draw glycine. Reason being, it's the simplest amino acids, and the R group is just a hydrogen. So instead of drawing R, you draw a hydrogen here. This is a skeletal structure. You can see that R is not drawn anymore. It's just amine group, carboxylic acid group, central carbon. Hydrogens are not drawn. This is a glycine. One more name to remember is cysteine. Cysteine is an amino acid that contains sulfur, and it looks like this. This is the R group, CH2SH. Now, you don't need to know how to draw cysteine, but you definitely need to know how to identify cysteine. So yeah, this is cysteine. And you can see here how there is sulfur as well as the skeletal structure, so it's quite easy to point out. You know, just look at the sulfur and you know, oh, this is cysteine for sure. Of course, there are some other amino acids that have sulfur as well, as you can see here, but cysteine is the most important one because it's involved in bonding. Okay, you will see later on something called disulfide bonds. Cysteine is involved, only cysteine is involved in making these disulfide bonds. Okay, anyways, glycine and cysteine, the only two amino acids you need to know. Now, what happens when we take two amino acids and put them together? They become a dipeptide, a dimer, a dipeptide. Now, dipeptides have peptide bonds. So yeah, don't get that confused. Dipeptide, peptide, polypeptide, right? Everything that has the word peptide in it, peptide basically means protein. So remember that. Now, how does the peptide bond work? Now, the peptide bond is formed between the H of the amine group and the OH of the carboxylic acid group. So in every condensation reaction, okay, in this case, you remove H2O and you join them together and you're able to form this C double bond O and H group thing. And this is actually the peptide bond. So this is con, I remember it. So C double bond O, and this tells you it's the peptide bond. Again, you need to know how to draw this and you need to know how to recognize this in diagrams. Now, of course, if you take many amino acids and you have the condensation reaction multiple times, this is going to give you a polypeptide. And the idea is the same. It's just that, you know, there are many, more than two. There's definitely more than two amino acids that are involved now. So these are 4 amino acids, and as you can see, H2O is removed from the same area. Okay, H from the amine group and O from the carboxylic, OH from the carboxylic acid group. Remove the H2O, join them together, and you get C double bond O and H. And this here is the peptide bond. There are 4 amino acids and therefore 3 different peptide bonds in between them. You can see in blue color how the R groups differ. And again, you don't need to know how to draw them. You need to know how to identify cysteine and you need to know how to draw glycine. That's all. That's the only two you need to know. Of course, you need to identify different molecules and different polypeptides. Sometimes they give you the skeletal structure, which looks absolutely scary now. But if you take a closer look, you'll be able to identify the R groups. Now, I have this animated, but I think it's easier if I draw it to you. If you want to try to do this on your own, pause this video and then play it. Pause this video now, and then I'll run it through with you when you play it again. So yeah, pause this now. Try to figure out where is the R group, where is the central carbon atom, so that you get to wrap your head around it. Okay, so first off, when you look at this, if they didn't label anything, you should already know this is a protein. And the reason why you know this is a protein is because, well, there's a lot of nitrogen. And the biomolecule that has nitrogen in this and has like this long kind of structure is probably a protein, like guaranteed protein. Okay, so it has C, H, O, and N, guaranteed protein. Now, how do you find the central carbon atom and the R group? Now, what I like to do is to look for the peptide bond. So the peptide bond always occurs between the carboxylic acid group and the amine group, right? So it has this C double bond ONH kind of structure. So this is 1, this is 2, this is 3. Okay, this is the peptide bond. And in between them, okay, in between this bonds is definitely the central carbon. Now, how about the ones at the end? Okay, so since this is beside this, so that will be the central carbon. Now, how about the R groups? Now, the R group is always connected to the central carbon. So we have R group 1 here, R2 here, R3, and R4 here. So this is a general protein structure. It doesn't tell you what is R1, R2, R3, and that's fine. We can see it in others as well. Okay, this is it makes it slightly more complicated, but it's not impossible. So again, we look for the peptide bond, C double bond ONH. Here, C double bond ONH. Here, C double bond ONH, also here. So this here is again the peptide bond. The carbon here between it would definitely be the central carbon. Carbon beside it would definitely be the central carbon. Okay, of course, at the end here, there is an amine group, which is not bound to anything. Okay, so let's just ignore that. This is just an amine group. This is central carbon. Now, where is the R group? The R group is here. This is the R group. This is probably a cysteine because it ends with SH, right? So this is probably a cysteine. This central carbon here, what is the R group? Now, you don't see any R group, and that's because H is not drawn. The R group here is H. Here also not drawn. The R group here is H, which means it's a glycine. And then last but not least, we have the R group connected to the central carbon here. Again, SH. So this is a cysteine. This is cysteine, glycine, glycine, cysteine. So yeah, that's how you identify the R groups. And of course, at the end here, there is an extra carboxylic acid group. Great. Let's move on to the last crazy protein, and you can see how it kind of looks complicated, but I assure you, just use the same method, right? Let's find the peptide bond, so C double bond ONH. This is a peptide bond here, there's a peptide bond here, and there's a peptide bond right here. Then let's look for the central carbon. The central carbon would be right beside it, here, 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 and here. Now, how about now the R groups? So what's connected to the central carbon would be the R group. This here is the amine group, you can see here. So this must be the R group. Here, nothing is connected. It's probably a glycine, so it's a H. Nothing here, so it's a H, so the H is an R group. It's a glycine. And then, how about next one? Here's the carbon, central carbon, right? This here, everything connected would be the R group. And last but not least, we see the last carbon connected to carboxylic acid group at the end, so not bounded there. So that doesn't matter. What matters is what else is connected to it. So the remaining one would be, again, the R group. Ta-da! And that's how you kind of break down the different parts of the polypeptides. If they ask you to circle all the R groups, now you can. Ta-da! Okay, now we gotta move on to other types of bonds in the protein. Now, as we said just now, throughout so far, we saw that polypeptides, dipeptides all require the peptide bond. Now, this is true. Peptide bond is found between different amino acids. It's present in all polypeptides. But other than that, there are 4 more bonds that you need to know about. Okay, these bonds occur throughout the protein, okay, and not just between adjacent monomers. Adjacent means beside each other. So not just with monomers beside each other, but like you can see here, it can fold and coil and bind like this. So there are 4 types. Number 1 is hydrogen bonds. Number 2 is disulfide bonds. Number 3 is ionic bonds. And number 4 is hydrophobic interaction. And we'll be going through them one by one. So let's look at peptide bond first. I will say that, as we said just now, it's because of the structure here, C double bond ONH is between the C of the carboxylic acid bond group, and N of the amine group. So this bond here will be the peptide bond, right? And as you can see, it's labeled here more precisely. C double bond ONH is a peptide group. So yeah, between C of COH and N of NH2. Now these are very strong bonds. They're covalent bonds, and yeah, they're very strong. They're not easily broken. And of course, they're a result of condensation reaction present in all polypeptides. Now, the rest of these bonds might differ between different proteins. But here's the first type, hydrogen bonds. Hydrogen bonds are actually individually weak, but many things can form hydrogen bonds, which make it cumulative strong. Now, hydrogen bonds can form between 2 different places. First of all is where the peptide group is at. So you can see here C double bond O and H, and here C double bond O and H. They're not beside each other. They are like opposite each other, but you can see how the double bond O, right, can do the hydrogen bonding with the NH group. Okay, so this is between peptide bond, between peptide groups, perhaps would be more appropriate. Not the peptide bond itself, but the H and the double bond all bounded to the peptide bond. Or this could happen between R groups as well. Okay, so R groups would be like this. So these are 2 different R groups, right? You can see how the oxygen and hydrogen would have some kind of bond as well, like this. So yeah, these again are individually weak, so they are easily broken by high temperatures and pH changes. So yeah, remember that it will become important later on. So again, hydrogen bonds, they form between the different peptide groups, like this, or between R groups, and they're easily broken. Next, disulfide bonds. Now, I mentioned this a little bit just now, and they are basically between sulfur atoms of cysteine amino acids. That's why cysteine is so important, and you need to know this amino acid because they are the only amino acids that can form this bond right here. This is a covalent bond. It is very, very strong. It's peptide bonds and disulfide bonds are about the same strength. Okay, they are covalent bonds. Number 3, ionic bonds. Now, ionic bonds form between different charges. So if one's positive or negative, that's going to attract, right? So it could form between 2 things. It could be 2 possibilities. One is the ionized amine and carboxylic acid groups that all amino acids have, and usually these are at the end of the polypeptide chain. They are not involved in peptide bonding. So at the end, probably ionized, so can interact. So yeah. Or ionic bonds can also occur between charged R groups. So as we saw in the list of 20 types of amino acids just now, some amino acids can be positively charged, some can be negatively charged. And if they are nearby each other, or they are coiled and fold, if the polypeptide is coiled and fold so that they are right next to each other, then that would cause them to have an ionic bond as well. Now, ionic bonds are weaker than disulfide bonds and as well as peptide bonds, which are covalent, but they're definitely stronger than hydrogen bonds. Definitely. Okay, but also, like hydrogen bonds, they're easily broken down by pH changes and high temperature. Sorry, I forgot this line. So hydrogen bonds, ionic bonds, both can be broken by pH changes and high temperature. Okay, fourth, which is the last bonding we're going to talk about, hydrophobic interactions. If you do chemistry, you might know this as van der Waals forces, but we don't actually use this term in bio. We use the term hydrophobic interactions. And the idea is that non-polar or amino acids with hydrophobic R groups, they're going to clump together. They're going to repel and move away from water. And as we said just in like previous lectures, I think, well, if they're the same chemical property, they probably will group together. They'll probably interact. So non-polar R groups tend to repel and move away from water and come together. Come together. So glycine, alanine, valine, leucine, methionine, and isoleucine, they tend to clump together away from the water inside the protein. So yeah, these are the weakest bonds though, because there's not really any bonds forming per se. They just interact because they have a similar chemical property. So if we were to, rank the bonds from weakest to strongest, you're gonna rank it like this. It will be hydrophobic interactions weakest, hydrogen bonds, ionic bonds, and then covalent bonds, which are disulfide and peptide bonds. So yeah, these are the bonds you'll be looking at. Now, why are we talking about bonds? Well, that's because, again, there are different ways a protein can fold. Now, between the monomers, between individual monomers, there must be peptide bonds. But then there are different levels of protein structure, and as they coil and fold, more bonds can be formed. So let's start from the primary structure. Okay, primary structure here is basically a sequence of a chain of amino acids. We're not talking about all the amino acids are the same. There are 20 types of amino acids, and it can be in any specific arrangement. It's a specific sequence. Specific is a specific way, meaning that it's properly arranged in a specific order, with specific types of amino acid, depending on the type of protein you're producing. This is a primary protein structure. It's not folded just yet. It's just a linear sequence. It only has peptide bonds. Now, how about secondary protein structure? So this could be in two forms. It could be coiling as an alpha helix or folded as a beta pleated sheet. Either coil or fold. So either alpha helix or beta pleated. And in this level of structure, okay, peptide bonds are present and also hydrogen bonds. Now, how about if it gets more complicated? So this would be tertiary. This is a 3D structure, and this would include coiling and folding, fold and coiling. And at this stage, okay, this structure is a specific shape. Okay, it's not like random. Like the amino acids from the primary structure, right? Since they're in a specific sequence, right? Which means there are different R groups there which can interact differently. These side chains, these R group interactions will result in a specific 3D pattern. And the R groups and the protein, within the protein, there can be all sorts of bonding. So it could be peptide bond. Peptide is always there between the monomers. There's hydrogen bond, there's ionic, there's hydrophobic interactions, and there's also disulfide bonding. And this is, so all the bonds will be present in the tertiary protein structure. Now, what happens in the quaternary protein structure? So quaternary protein will also have all the bonds, just like tertiary protein structure, but with the addition of more than one amino acid chain. So far we're just talking about one polypeptide. This is more than one polypeptide. So before we move on and look at each level with more detail, I just want to say that polypeptides, they are made as a linear chain first, and then later on coil and form into the specific structure. And different proteins would have different levels of structure. We'll do a few examples in a moment. But yeah, again, let's go through each and every level, and we're going to define them. Primary structure is a linear sequence of amino acids. Again, we are talking about a specific sequence of amino acids with different properties of R group. And this determines the folding of the polypeptide chain later on. These different R group interactions will determine how it folds later. Okay, so all amino acids do this. All polypeptides have this. Now, secondary structure, okay, some have, some don't, but every polypeptide is capable of this. Why? Because these are hydrogen bonds between amino acids, and honestly, every peptide group can form hydrogen bonds, right? So every polypeptide is capable of having two kinds of formation. Number one is alpha helix or beta pleated. Alpha helix is when there is hydrogen bonding of an amino acid with the amino acid 4 places ahead. What does that mean? Okay, assuming that this is amino acid number 1, okay, it's gonna bind with the amino acid 4 places after, right? So every 4. So this is 1 amino acid, 2, 3, 4, 2, sorry, this is 1. So 4 places ahead is plus 4. So this is 1 plus 4, so it has to be 5, 2, 3, 4, 5. So you can see this is binding to this, which is 4 places ahead, and it repeats like every 4 amino acid it will bind. So here's 5. So let's continue from there. 6, 7, 8. Oh, I missed one somewhere. 6, 7, 8, probably behind that we cannot see, and then 9. So you can see that this feather binds with this feather, which is 4 places away from it. And if every amino acid does this, then it would form a spring-like structure, a coiled structure. How about beta pleated sheets? These are hydrogen bonding as well. And as you can see, it's a slightly different conformation. It's still between the peptide groups though, as you can see here. Okay, like this or like this, right? And what will happen is that it will form hydrogen bonds with the opposite one. This is a straighter, looser form, but basically they have parallel folded sheets like this. So yeah, that is secondary structure. Tertiary structure. Now this is both coiling and folding. Just now it was coiling or folding. This is both coiling and folding in the same polypeptide chain. And this is coiling into a precise 3D structure. And as we said just now, it's due to interaction between R groups, and all 4 bonds are involved here, okay, and it would form like a nice little thing here. Now, an example of tertiary structure would be myoglobin. This is a protein which carries oxygen. It's present in muscle cells and give it a red color. Imagine like beef. Okay, beef has a lot of myoglobin, that's why it's considered a red meat. It appears red. And yeah, it's made out of one polypeptide chain only. And as you can see here, it's a very cute 3D structure made out of coils and foldings, and has even a heme group, which is a non-amino acid group, which can bind oxygen in the muscle. Now, this is only one polypeptide. And as you can see, it's a precise structure. It's not random, it's a precise structure. And this is because of, again, the specific sequence in the primary structure. Now, quaternary structure is pretty much the same as tertiary, except that two or more polypeptide chains are involved here, and you can see two examples here. Collagen, which is made out of 3 different polypeptide chains, and hemoglobin, which is made out of 4 polypeptide chains. And this is different from myoglobin, right? Myoglobin is just 1. This is 4 polypeptide chains. And therefore, it's considered a quaternary structure. So, this is a past year here, and the question asks, which levels of protein structure are demonstrated by hemoglobin molecule? And because of what I just said, you will go like quaternary only. And the real answer is this, right? The real answer is all 4. Even though it's at the quaternary level, but it demonstrates all 4 levels of structure. Why? Because it does have a primary sequence, a primary structure, which then coils or folds, secondary, and coils. This coiling and folding results in a 3D structure, and the combination of more than one polypeptide chain results in a quaternary structure, and therefore it demonstrates all levels. It's at the highest level, but it demonstrates all levels. And this differs, again, based on the protein. Not every single protein is quaternary structure. Like myoglobin is just a tertiary level, right? Some proteins are secondary structure. So yeah, it really, really depends on the protein as well. But yeah, for this hemoglobin, definitely 4 levels of structure. And yeah, that's the end of this video. This is part 1, technically, of the protein video. Next video, we're going to explore what are globular and fibrous proteins and learn 2 examples. So yeah, that's it from me. I'll see you next video.